Молекулярный биолог, научный сотрудник,
molecular biologist,
Харитонов.
research associate from Moscow State University, Sergei Kharitonov. But I'm sorry, Sergei, please welcome to the stage. You have the floor. Do you need the shades? No, I'm good. I'm good.
Харитонов, МГУ.
Sergei Kharitonov from Moscow State University.
Спасибо, привет, друзья.
Hi, everyone. Thank you, friends.
Спасибо, что дождались.
I'm glad to be here. Thank you for staying here late with this
Может быть,
heat.
впереди меня еще ждут такие регалии. А мы с вами тем временем...
Maybe what I just heard right now will be in my future, but we continue to talk about AI, one of the bigger topics, and we'll talk about how AI is integrated into medicine, what kind of opportunities we have in the nearest future. Because we will be dreaming, we will be thinking about potential things that might happen in the future, what we want to have in our pocket. But at the same time, we might say that, okay, we have some of them already right now. Some of them are used in pharma, some of them are used to cure and treat previously untreatable diseases. So AI in medicine.
Импакт от внедрения такого
I
мощного...
would say that the impact of the implementation of AI to medicine will be enormous.
С тем, как в 20 веке
It can be compared to the 20th century antibiotics, and you can split the history
было...
on this moment because people died from scratches, and it was basically a capital punishment. But now we have good medication. You might have some troubles, but you will not die from it. People died before invention, before these antibiotics. So AI might be the same degree of shift in terms of medication we get access to, and in terms of longevity, how long people can live and the quality of life. But can it be different? There is an apocalyptic scenario where AI might result in catastrophic mistakes from the medical industry, and we will talk about this potential two excluding scenarios and how AI is implemented in this industry. But first I would like to learn, why do we need AI in medicine? And even if it's very complicated, even if it's very difficult, people will still use AI. The first reason why we have algorithms in the medicine is because there is a significant increase of data that are stored in biology, biomedicine, different adjacent areas. There's a genome data, protein data, and all those OON data on how humans are constructed, how animals are constructed, what kind of genes, mutations we have. Those are terabytes and petabytes and xenobytes of data, and it's stored every single day. Previously, imagine you are a scientist in the 19th century. These people, if they are pedantic, if they are really good experts, could have all the literature on their industry. They could actually know everything that was known up to that date. But today, just if you learn, there's PodMed web resource, and there are 30 million publications and about 16-18% dedicated to oncology. From 30 millions, that's a lot. That is a lot. But obviously, you do not, even if it's a very good oncologist, even if you work this adenocarcinoma, you cannot analyze all that information, so we need support.
That means we need some support to at least identify the gap. That is the gap that really highlights the clear opportunity for a new discovery. So the researcher that reads articles for the new research is actually reinventing the wheel, because he does not know if it is an innovative study or not. I mean, it's hard to understand this by yourself single-handedly, and you do need some support. So you need the systems to analyze the natural language that will make it possible for us to digest thousands of articles that come out every year without spending all your working time into this. Now, a whole different goal is to understand what are you studying. If you're a molecular biologist like myself, you research into the genes, you analyze the DNA, you read the genomes. So in order to analyze the data from your own experiments, that is, to understand what you have actually done there, you need artificial intelligence. I mean, if you read DNA, in 1977, the lab led by Frederick Sanger published the first method of genome reading, the DNA reading, or the genome sequencing as we know it these days. That was a real breakthrough. That was a piece of agarose gel. A staff member of the lab would read it, sequence it visually, single-handedly, refer to a file, and then read the 500 to 800 nucleotide. The letters of our genome would take like up to a week, and a whole lab of researchers operating. Our genome is 3 billion nucleotides. You understand this volume, right? Sanger, the top-most expert in this, who gave the name for the sequences, in order to read the little genome of a little bacteriophage, that's the one that infects the bacteria, that was Px174. For those interested, its genome is 5,400 nucleotides. So they spent 5 months reading this genome. I mean, it's no good. I mean, when you do your own reading, it's extremely inefficient and it takes a long time. So you clearly need some support now. Today, we have highly efficient sequencing, and that's what I work with. They produce 10 times more letters of nucleotides the reading capacity. No lab will ever be able to compete that, and to beat that, we certainly need artificial intelligence. We need machine-based methodology because we need to read the data and we need to interpret the data. We need to make sure we find the complex trends that will then come out as new technologies and as a new understanding of how we work as humans. So the implementation of the machine learning as deep learning and artificial intelligence into the genome reading and into biomedicine is inevitable. I mean, for purely scientific research readings. So that is not the only reason. There are also some economic reasons to it. You've got to understand that not only the researchers need artificial intelligence to understand what exactly they're doing. Medicine and pharmacology is a huge business. Business is there to make the money. So in order to imagine what it takes to develop a drug, just take any working drug, it usually takes 8 to 15 years of effort of several teams, including several universities, and it's up to $3 billion. I just don't want to get the zeros wrong, but it's about $3 billion in expenses. It can be tenfold more, tenfold less, but roughly that's where we stand. So the big pharma, as the initiator and the motivator of accelerating this process, is a real pioneer in implementing artificial intelligence in medicine. And there are lots of cases we're going to discuss today. They come from here. They've been doing it for a very long time. They know how to do it extremely well. And now we have the new ideas that have been generated for clinical trials by artificial intelligence. Now, let us see how and when this works, where and how artificial intelligence can participate in drug development. I mean, of course, artificial intelligence and broadly this deep learning, the machine learning and the algorithmization of research is involved in drug development at all stages. That is starting from the analysis of literature to the data formation to the formation of the design and the criteria of patient enrollment into clinical trials. I mean, you have the molecular screening, the molecular design, the toxicity forecast, the optimization of intake modes. So all the models available today can partake in this and be very efficient. Let's see what comes inside there. In the first place, you do the disease analysis, that is to treat a disease. You need to start with searching for a target, not the drug. You need to understand what's going wrong in the body that you have to treat. It's either a protein or a gene. It's an enzyme, a receptor. A clear-cut example would be The genetic disease that's very well known, it's the cellular hemorrhagic anemia. It's where you have a disruption in how the protein carries the oxygen in erythrocytes. So within one protein, just one little element breaks, and the erythrocytes change their shape, then the oxygen supply changes and the metabolism changes, and the only solution is the hemoglobin gene. If you can adjust it through gene therapy, we will be able to treat this disease entirely. So if in case of this type of anemia, it is easy to adjust just this gene and everything will be fine, it's not going to be as easy with drinking, with heavy drinking, because it's not a monogene disease. It involves several thousands of genes. Some have to do with the motivation system based on dopamine. Some have to do with alcohol metabolism. What is the target here? I mean, to write down all the genes on a sheet of paper, you still spend quite a long time. I mean, it's so different from the sickle cellular anemia. So you've got artificial intelligence to show you here are some genes, the correlations possible are such, and this is the most promising thing for a commerciant. I recently commented in the news, a group of Russian scientists identified this gene and demonstrated how this gene is responsible for the development of drinking syndrome. Now, this is related to the neural networks. Now, you not only identify the target, you also in quantitative terms characterize the connection of this target with your disease, with the pathology you're working on. I mean, if you act on this target, how is this going to affect the disease? You've got to know the figures, otherwise you will not be able to predict the result. And the neural networks do this as well. One is natural language processing. That's the neural networks that read the research articles and look for trends in data, and it's possible the drug has been discovered, but as a human, you will not be able to find this information. The next part is graph neural networks. It's the neural networks that create the multidimensional graphs and connections between reflecting the correlation of different molecules in our body, and they identify the causal links. The cause and impact links. That is, if I impact gene X, what kind of correlation am I going to derive from that? These neural networks are being developed. There are the companies in the market that use these approaches to develop drugs in oncology, in virus control, in genetic disease control. So it's about analyzing the target. So with the assistance of the neural network, we first identify what we're treating, and then we move on. Next, we identify what we are treating. How do we treat this? What molecule do we take to get it to act on it? So in the first place, we get the broad screening trials. When you have a scientist, a researcher taking like 100 mice, 500 battery plates with different types of cells, affects them with all the molecules that should work on them, sees what happens, and then selects the right candidate molecule. It's very expensive, it's time-taking, and it's complex, and it's not necessarily predictably positive, especially when it's about the new diseases we hardly know anything about. Artificial intelligence today analyzes the databases of the well-known protein structures and the databases of the plant-based extracts and the existing biological molecules can predict affinity. That is the correlation between the molecule and the target. So artificial intelligence that you have in all the databases will immediately select for 50,000 candidates we would never be able to identify by ourselves. Then he runs them, artificial intelligence runs them through the set of criteria. Some get discarded because they're not absorbed. Some get discarded because they're not stable. So we get 12.5 out of 15,000. And then using neural networks and algorithms, we come up with 100 molecules that are impeccable, and we take them into research on living organisms, and that's going to be much faster and much more efficacious. We don't have to spent 5 years to look for like the first 100 of good candidates. We will only invest like 48 hours for good pipelines. The next part is optimizing the properties. We know that this molecule works against the disease we're interested in, but it is not perfect. The generative models and neural networks could offer the modification of a network so that it can acquire the good properties like acetyl salicylic acid did not come out in this composition right away. In traditional medicine, they used the bark of salix or salice in Latin. They would make a brew, and this brew would contain salicyl. I mean, indeed, salix is the willow, right? And it will generate salicylic acid. They realized it was an acid. They called it salicylic acid. It was good against the fever, but it was not efficient enough. So some chemists came out with adding 2 more carbohydrate atoms and adding salicyl there, acetyl there. So he was a chemist. He did not know how that was going to go. It did not necessarily have to work out. And actually, aspirin today is not produced from the salicyls, from the willow. I mean, there are no willow forests anymore. It's all synthesized from oil. If you take an aspirin pill, it's a petrochemical, it's an oil product. So optimizing the properties. And thirdly, it's the de novo molecular design. That is when we do not know what it is all about. We don't have a molecule, we don't have an idea. So we go artificial intelligence, you go and synthesize it. So today, the generative chemical models devised in the pharma can propose a completely de novo structure based on their own dark laws that are not known to us, that will have the right properties we need, and it will optimize the chemical synthesis methods so it's easier for us to adapt it industrially. It's an amazing story, saving dozens of years of development to everyone working in the pharma, and outside of it, it's very powerful, like really big thing. So we have now identified the molecule, we've done the screening.
Вообще, несмотря на то, что мы уже верим в эффективность этих кандидатов,
now we trust the efficacy of all these candidates. Now, it would be great if we could understand if they link to anything else in our body. I mean, are they poisons? Are they venomous? Well, the patient dies, he no longer complains, right? So job well done. But there are some families surviving, there are some kids surviving, they may not be happy with it. So check out the toxicity. That's why we have neural networks. Before that, the colleagues have discussed the metabolic duplicates, or doubles. I mean, it's the complex graph neural networks that describe how our body works from inside. You can upload into the system more molecular candidates, and you can check if there is any cross-reactivity there, if they kill our cardiac muscle, if they kill our liver, if they are mutagenic, if they are cleared by the liver successfully. In fact, antibiotics and many drugs used to be mutagenic. Today, you can just check it out on your computer without leaving your lab, and you don't need to waste thousands of lives and years and money to develop the drugs that are not going to work in a focused way. Toxicity, we've ruled out the toxicity. So out of the 100 non-toxic candidates, we have 50.
Это, конечно же, прогнозирование
Next, we predict pharmacokinetics, and we do some more precise prediction of efficacy, efficiency. Every substance we predict to work has to be absorbed in some way in the body. It's got to be spread in some way inside the body. It's got to go through the metabolic changes. And also the release is important. I mean, how is it cleared from the body? Is it through the kidneys? Is it through sweating? Does it physiologically get into the physiological liquids like the salivas and others? You need to understand all of this about any serious drug that has side effects. All good drugs have adverse events, because there are the effects and there are the adverse events or adverse effects. I mean, that does not work differently with the drugs. So the neural network today evaluates both the absorption locus and the biochemical route of absorption. Next, they can predict if the substance is spread evenly in the body or if it gets accumulated in some tissue. Methylene blue is a very well-known dyeing agent in the narrow circles, schizomedics, as we call them. It's a drug against all the diseases. It's been used against tantrums, drinking, schizophrenia, epilepsy. They used to use the methylene blue to treat all of this. So the methylene blue, when drunk, is accumulated in the brain. It actually doesn't spoil the brain. It actually makes it better. But when you do an autopsy, the brain is going to be very strongly blue, dyed in blue. So unlike the methylene blue, that is not very toxic. Some substance, when it gets accumulated in some tissue, can become toxic, like, for instance, mercury. The mercury we eat all the time with the seafood, in those doses, it's not toxic. However, if you eat the shrimp and the mussels that filter the water, that's where you get most of the mercury. And if you eat it all the time, you get quite a lot of mercury in your body, and you accumulate it in your bones. Generally, the body is fine, but the bones may be getting worse. By the age of 60, some things that are not there, it doesn't may well happen. So the idea about the spread, distribution is very important, because no reasonable clinical trial, no contemporary clinical trial will demonstrate it to you. I mean, you've got to get the drug to the market sooner to make at least some money on it, because you cannot just waste $100,000 and not learn anything. Now, hallucinogenous fungi, you get it into the body, it gets cleared, nothing changes. But many drugs, in fact, get transformed. So we get an additional job here. We need to evaluate what are the interim products that are created as part of our metabolism as we process the drug. So we get it into artificial intelligence, we analyze metabolism, and you see that alcohol, for instance, spirit, is toxic, and it has acetaldehyde. That is very toxic, and it's a byproduct of alcohol. It's linked to DNA, to proteins, to sugars. It disrupts the enzymic function, and there is no good about that. In fact, they even have the radiomimetic effect. That is, it contributes to the creation of the adverse mutations, especially in the epithelium, or the spermatozoa also is affected because the quality of sperm goes down. I mean, you've got to understand the particularity of this metabolism, and without artificial intelligence, it's really hard to get it known. Now, the clearance, kidneys, saliva, I think you know where I'm driving. Okay, we understand now everything about the candidates we have. We're still sitting in our room, in our reception. We're using our computer. We are using the capacity of some server. We just mouse on the screen and do these large-scale studies. We understand we now have 20 candidates left. So we start researching into them in the live models. So initially, the new
Сначала новое лекарство нужно подтвердить инвитро. Это называется в пробирке, когда берут какие-то культуры клеток целевой ткани, на которую должно подействовать лекарство, поливают его этим лекарством и смотрят, а что будет, да? Ну, то есть клетки как бы помрет или не помрет, скукожится или не скукожится, как вообще пойдет дело, вылечится, изменится ли метаболизм клетки в лучшую сторону от того, что мы на нее накапали. Это вот эти инвитровые исследования. И тут что важно понимать? Вот тут уже начинается работа, где нужна прикладная деятельность человека, всегда была.
And here we have in vitro activity where you need actual humans. So, for example, you do a few drops, then you look at the tissue, and if it died, then it's a simple dichotomy. But there can be more complex, some morphological changes.
Otherwise it may change. This means you can look at this tissue and you can immediately understand that something is going wrong with it. I mean, in fact, you've got to be very, very well versed in understanding visually what a healthy cell is like, how granular it is, what the nuclear size is, how it moves, and what the pegs it uses are. So you have to understand all kinds of different things about the cell. Now, if an expert is not really While TNN today is not very active, he will not be able to notice the morphological changes in the target drug, and that's bad. So here we have computer vision to assist us. We train our model using a lot of visual data that are correlated, that are duly collected and prepared. So in this way, our neural network learns to understand all by itself single-handedly which of our cells are healthy. So in silico, that is on the computer screen, we just do a whole range of phenotypic screening. That's the phenotype analysis. So that enables us to check how the looks of the organoids are, and this makes it possible for us to make a lot of predictions. So I'd like to share one case with you. It's about computer vision. And in fact, it's interesting how it is used in reproductive technology. I have a good close friend, Anastasia Sisoeva, who is a clinical embryologist at the Kulakov Center for Obstetrics and Gynecology. She has fertilized these two embryos with her hands in the vials and took a picture of them. So Anastasia Sisoeva can now tell me which of these two embryos is going to fit well. That's like a beautiful little embryo that it will very soon be transplanted to the uterus, and which one is not really excellent, so it may be better to implant both. She sees this, although morphologically, in terms of what the books say, both embryos are perfect. They're good, they have all the properties required for implantation, but her experienced eye, her live neural network sees that one is more granular, it looks more powerful, the other one looks less powerful. You can look at them and find those 10 differences. Let me tell you, the ones who voted for, okay, just take a second. Which one do you choose? Which of the two embryos is going to be more implantable? The green one, the one with the green smile, is better. It's the top one. It's very powerful, super powerful. This one is not. An embryologist will never be able to explain it to you. It's just a matter of intuition. I mean, all these intuition-based things are optimized by phenotype screening algorithms and networks and artificial intelligence. And in fact, the computer can detect this much sooner than an embryologist. I mean, you won't have to implant like 6 embryos hoping that one will work out. And that means you will be able to do the simulation with lighter hormonal protocols. And this IVF implantation is going to be much easier for those women who go for it. Several months ago, they had the sperm races in Los Angeles. Indeed, they were 2 racers, so their samples were taken, they had a race, and the sperm cells, their spermatozoa, really had to race to reach the target. I believe for reproductive supportive technologists, this was great. Where you have the races, you have the bets. Where you have the bets, you have a lot of money. Where you have a lot of money, technologists are going to follow suit to catch up. There will be a new race out. There is a new artificial intelligence option very soon that will just take a photo of the racer and know if he's going to produce excellent embryos or not. In fact, the embryologist cannot write up the neural networks, but otherwise, this is where it's going to flourish. In Zone F, I'm going to be available in the collapse zone. Now, please come see me. So, next, we now come to the point where we get the greatest responsibility. We have 100 excellent candidates that are computer models. So we sieve it through to 10 to 12 candidates that are not toxic, and it works fine. Now we've tested them on these cells. These cells are not laying out. Everything is fine. So we go into trials in humans. And again, it's difficult to do single-handedly because we need an adequate design of a clinical trial that would produce a very good informative outcome. It will not be too expensive, and hopefully no one dies because it's real people, that's difficult in terms of ethics. So this takes a certain selection of patients involved. So you've got to select the patients who do not really suffer of the intake of this drug. So you analyze the patient biomarkers, the stratification to do the right regimen dosage. and even the forecast of the patient's response to therapy, all of this is feasible with metabolome models. But in labs, this works. And I'm sure very soon some big things will be done in this area in order to manage some very difficult, complex orphan diseases, or, for instance, for cancer. So the pharmaceutical companies have a huge wealth of work to do. Drug development is complex, and you now understand how it all works with Zolgensma, or with the spinal muscular atrophy drug that is well known for its high cost. It's the endogenous therapy that corrects one gene that works in the wrong way in the bone marrow and causes the muscular atrophy. It's something that can treat a rare genetic disease that used to be a death sentence. Now you can just infuse it once. You had to have drugs to take permanently. The cost would be about $500,000 a year to sustain a patient like this. I mean, it was really extremely expensive. Now, the guys created the therapy. They created the gene-based drug based on paradenovirus, and you just need one infusion to treat the patient. It's a 60-minute infusion. It's worth $2 million. It was in development for 23 years, and then they calculated that the 10-year support to the maintenance is worth $10 million. So if they do an infusion at the cost of $2 million, they save $8 million, and they can set the prices as high as that. It's the second most costly drug. I cannot remember the first one. It's to work against Duchenne muscular dystrophy. That's another very rare orphan genetic disease. It's worth the gene therapy is available. It's 3.5 million thousand per one injection, and you see each of these stages can be either automated or simplified or reduced into just several days instead of working on it for years and decades, thanks to machine learning and artificial intelligence. Well, this is a real revolution, and it's a breakthrough for people with orphan diseases. Why? Because if you have an orphan disease, it's of little interest to pharmaceutical companies as a business. So if you've got to invest 23 years into exploring a drug against the disease, bringing together like 150 individuals in the world, like this whole Gensma I was talking about, the spinal muscular atrophy drug, it doesn't really make sense in terms of a business model. So traditionally, they do not invest into this as a business. Now, with the introduction of artificial intelligence, the situation may well change because you will be able to do it faster. You have 7,000 orphan diseases in the world. Most of them are unknown. Few cases, little investment into the research, but this can change also. There are other diseases that are extremely common. It's cancer, all kinds of cancer. You understand that cancer is like thousands of diseases coming under different names. I mean, oncologists do not know of cancer. They have the squamous cell carcinoma, they have the small cell carcinoma. I mean, I'm not an oncologist. I just know all these names from Dr. House of the pheochromocytome. But there are thousands of diseases of very different biological origins and different in terms of metabolism and approaches to treatment. There will never be in one drug against cancer. Now, developing one per every type, well, that might be a promise. We seem to be handling it gradually, but the multifaceted diseases are a whole different story because it's a diverse range of diseases you cannot just tackle overnight. But using the graph-based neuroworks, we can certainly find more universal solutions. And bringing this all together, how can we hack the orphan diseases, all the system of the difficult development to find a key to an orphan untreatable disease? In the first place, that's accelerated diagnostics, gene, 300 different syndromes. They taking, for instance, the Down syndrome. They diagnose 300 syndromes from a photograph. In order to set a proper diagnosis, it takes ages. It takes years because it's unclear symptoms. So the neural network just takes a photo to set a clear diagnosis. That's better treatments, that's better socialization, that's accelerated outcomes. And then the search for the targets. Here you have the Insilico Medicine case, an excellent company with a Russian founder. It's an American company, though. They have now created a drug for idiopathic lung pulmonary fibrosis. When you have this word, idiopathic, it means the doctor does not know the cause of this disease. Idiopathic is coming from nowhere. God knows how it developed. The pulmonary fibrosis, we do not know how to treat it. We have supportive therapy. We do not know what comes out of our treatment. So they created a drug. They found the target. They came up with the idea. They did the preclinical stage through the Insilico Medicine neural networks in 18 months instead of 5 years. 18 months got the drug into the second phase.
And this is an absolutely amazing scale for previously untreated diseases. For many people, this is a ticket to life. And finally, there's a company that's called Benevolent AI, and they use this artificial intelligence graphs with billions and millions of connections between genomes and diseases. And what they do, they took the COVID medication, they took the arthritis medication. WHO, World Health Organization, introduced this, and it helped many people to save their lives. And I think from the side, sclerosis, one of the neurodegenerative diseases which kills you slowly and you can't really treat this, but they were able to create a working molecule in 12 months, 1 year, not 5, but only 12 months, not 5 years, only by using all those smart systems and AI. So it is evident that the potential is endless. It is here. Those are pinpoint cases. But for each of us, for each of these slides of this presentation, you can get examples from Russia, from Europe, from the United States and other countries too. And finally, the icing on the cake. These are the dreams, this is the future. When we have these digital twins, what we were talking about previously, when you can come to a doctor, you take a scanner, you give the analysis, maybe your blood or your anything you want to be analyzed, then AI creates your metabolic twin, and then a battalion of viruses and microbes will be tested on your twin, and then you will get a cocktail personalized for you, the molecules that will actually help you, your body. It will increase your longevity, it will help you age, help you with health, improve your mood. Oh, nothing bad, but only good, but you do understand what I mean. And finally, there will be targeted therapy, oncology. For oncological patients, they need special medication because, as I said, there are different cancer oncology. You have different unique mutations and changes. Usually, you obviously want a single silver bullet medication, but usually you need a custom-made medication for that specific patient, and you can improve the speed for the production using these smart neural networks. So there are a lot of great things, and unfortunately, I'm running out of time. But I would like to cover a single case. It's absolutely amazing one. When the artificial intelligence, it's not about medicine, but AI helped find the connection which completely revolutionized one of the areas. In criminalistics, there was a law that you have fingerprints that are unique, and any police officer will tell you that they are completely unique and you cannot treat them. But this law was broken. AI analyzed thousands and millions of fingerprints, and it took into account the patterns that were not considered by people at all. There are differences between different lines of those fingerprints. two different lines, zigzag lines, and if you have a tangential line to those zigzag lines, then there is a certain pattern for angles, and you can see the same pattern for different fingers for the same person. And just imagine how many dead cases can be revived and they can actually be solved. So people thought that you have different fingerprints and different fingertips, but actually no, you can have the same pattern on each of your fingers. So there are still risks. You're not sure who is responsible. If AI is at fault, who is responsible for that? Somebody dies. Was it AI? Was it the pediatrician, doctor, government? You choose, you name it. It's not clear who to blame, and based on who's to blame, we will adjust the technology and adjust the walk to the future. So it is really, really hard. There are so many philosophical questions here, but we should remember this. There are so many developers here, people who work with ML, deep learning. So I call upon you, please go to medicine. It's a very interesting area, but we do not create just AI. We create the correct culture working with this very complex and multifaceted tool. So this is all I wanted to tell you. Thank you very much for the listening. Thank you very much for the attention. I have my Telegram channel. I'm talking about medicine technology. We have a few minutes. I wanted to ask you a few questions. You did not cover this in the presentation. Digital twin, that was my question. Well, okay, so once I get the patent, I will make you the co-author. And you said you would need millions of terabytes of data that you would do every day. So maybe not in 30 days, maybe not in 30 years, maybe in 5 years. Okay, but we will need computational power. Second point, we both here for the third time, given the presentation, listening to presentations, the brain will learn those predictive patterns. And I know about this. I actually took a look at you and I called you candidate of science, but you're not the candidate of science. I should be a candidate of science by the end of 2025. And just after he becomes the candidate of science, he will get his PhD at the same time. What you told us here in 40 minutes, this is so inspiring. My surname is Golubov, which is dove of peace. But actually, you are the symbol of peace. You are the symbol of harmony and bright future. Obviously, we have these professors from Harvard. We have Yuval Noah Harari. But you are the person. You are the beacon of light. You cover this gap. You give us the hope, together with AI, that we can fix our biological bugs. And thank you for Kulakov. And thank you for Karelia. Thank you very much. I'm from Karelia. I spent 20 years there. I do not recommend, but I do remember. Obviously, Joe Rogan and Mel Gibson would thank you too. This is what was in their conversation. No, you should not. You should not listen to them. You do not drink those supplements. And you should remember, those could be deep fakes. Maybe Joe Rogan would tell you about these supplements for hours, but it was a deep fake. Who knows?